Hello, everyone. Today we will be talking about your qualitative analysis of amino acids. So, this is a post lab discussion of your experiment 6. Our objectives for this discussion will be to demonstrate the properties and solubility of amino acids and enzymes using different qualitative tests, determine the solubility of some of the given amino acids, and to identify the amino acid present in each unknown sample. To start it off, so what are amino acids? These are monomer of proteins, or simply amino acids are the building blocks of proteins. So each amino acid has at least one amine group and one carboxylic and functional group. So the different properties result from the variation in the structures of different R groups, or also known as your side chain. Okay, so first we have your solubility test. So ang principle neto is that polar amino acids are more soluble in water than non-polar amino acids. So it is due to the presence of the polar side chain, which enables amino acids to accept and donate protons to aqueous solution. Okay? So dito, nadidetermine natin na miscible ang mga amino acids in water, chloroform, and your sodium hydroxide. Pero, di ba non-polar ang mga amino acids? So, how come they are miscible to polar solvents? So, we have this thing called Zwitter ions. So, based sa lecture ninyo, all amino acids have Zwitter ions. And this Zwitter ions, meaning, kung may ganito siya, ibig sabihin, capable siya of having your positive and your negative charge. Okay, so ang amino acid mo, meron siyang NH3 and COOH. So, sabi niya, meron siyang positive and negative charge. So, yung carbon group mo, meron siyang NH4 na positive and COO negative. Okay? So, ibig sabihin neto, dahil meron siyang positive and negative charge, nagkakaroon siya ng strong attraction sa polar molecules. Okay, so ano ba yung mga polar molecules ulit? Ano yung polar solvents natin? Your water, hydrochloric acid, and your sodium hydroxide. That's why nagiging miscible ang result niya with them. On the other hand naman, you have your chloroform, which is a non-polar solvent, nagiging immiscible siya doon. Okay? So here are the examples of your amino acids. So tyrosine, tryptophan, proline, methionine, arginine, histidine, glycine, and the others. So they are all miscible again with your polar solvents. Water, HCl, and your sodium hydroxide. So sa chloroform, bakit immiscible kasi siya? Dahil ang chloroform ay isang non-polar solvent. Now let's move on to the other qualitative tests. So this identifies the amino acid present in each unknown sample using different tests. Now first we have your ninhydrin test. So your ninhydrin test or your triketohydridine hydrate is a chemical used to detect your primary amino group. So ibig sabihin, yun din yung reagent mo. Okay? So, your result would be your blue to purple solution and a yellow solution. So, all amino acids react to a blue to purple solution, while proline is the only amino acid that produces a yellow solution. Okay, so ano bang nangyayari sa ninhydrin test? So, you have your ninhydrin. So that is your reagent plus yung primary amino acid mo, it would result into your hydrindantin plus ammonia. Okay, ito yung nagiging product niya. So kung mapapansin nyo, yung hydridantin and yung ninhydrin, So, it's almost the same. So, ang ammonia, papagitna yan dun sa compounds. Therefore, creating your Rumans purple. Okay? Reumans purple pala, sorry. Okay? So, sa Reumans purple, yan yung nagiging color ng solution mo. So, yung presence ng primary amino acid mo when combined with your ninhydrin reagent, nagpuproduce siya ng purple color. So, ano bang nangyayari? Bakit yung proline hindi siya nagpo-produce ng purple color? Because your proline is a secondary amino group. Again, your ninhydrin, when mixed with your primary amino acid, dun lang siya nagka-create ng ammonia. Dun siya nagpa-produce ng byproduct na ammonia. Therefore, producing a purple solution. So again, yung proline, pag mix siya with your ninhydrin, babalik lang siya into amine. Kasi ang magiging product niya is your hydrin-dantin plus NH2 only. Okay? Wala siyang ammonia. So in here, you have your first test tube. Yellow yung solution niya. Therefore, pertaining na it is proline. Bakit ulit? Kasi ito yung secondary amino group ang proline mo. Okay? While yung second test tube mo, it's blue to purple solution. Okay? Because ang primary amino acid na kakaroon siya ng byproduct na ammonia. Therefore, again, creating what? Your Reumans purple. Okay? So here, all amino acids except proline ang blue to purple solution. And proline is the only yellow solution for ninhydrin test. Okay, so first you have your xanthoprotein test. So dito, you detect your aromatic amino acids or it is simply your nitration of aromatic amino acids. Okay? So ano ba yung reagent na gamit natin dito? These are your nitric acid and your sodium hydroxide. Anong use ng nitric acid mo? So your nitric acid is your primary nitration product. So, ito yung nitrate sa benzene ring para magkaroon ng nitro derivative. Okay? Then your sodium hydroxide naman, it is used para mas maging favorable yung solution by increasing your pH. So, ginagawa niyang more alkaline yung solution mo para ma-intensify yung color. Okay? So in here, you can see that yung tyrosine and yung tryptophan, nag-change siya into yellow to orange solution. So it yields a positive result and then yung negative result naman is for your phenylalanine. Okay? Bakit ano yung phenylalanine? Bakit hindi na siya nag-react? So in here, yung phenylalanine mo kasi, yung aromatic ring niya, there are no substituents. Pure yung aromatic ring mo. So, it is stable na. Okay? Stable na siya and no need for exchange of molecules. So, in your lecture, di ba, yung benzene rings with resonance, they are so stable that hindi na siya ma-nitrate even if yung solution mo or yung environment niya is very favorable for nitration. Stable na siya eh. No need na for something to change with its compound. Okay? Okay, so next we have your histidine test. This is your bromination of histidine in an acidic solution. Okay? So histidine are brominated in acid solution followed by the neutralization of acid with excess ammonia. Okay? So, paano nangyayari yun? Ano ba yung reagents na gamit natin? So, yung reagent na gamit natin is your bromine water, your acetic acid, and your 5% ammonium carbonate solution. Anong use ng acetic acid? Your acetic acid decreases the pH, therefore, magiging acidic yung solution. And then, yung 5% ammonium carbonate solution mo naman, ini-increase niya yung pH mo or simply, it is your neutralizer. Pang neutralization siya. So, bakit gano'n? Bakit nagkokontrahan sila? Yung isa ginagawa ng acidic, yung isa ginagawa niyang neutral naman yung solution mo. Ano bang nangyayari? So, ang histidine kasi, it has a side chain which is called your imidazole. Okay? And your imidazole, specifically, may pH siya of 7. What does that mean? So, a pH of 7 meaning it is neutral. So, yung pag-change ng pH mo or pag-decrease using your acetic acid, mas madaling pumapasok yung halogen mo, specifically your bromine sa amino acid. Okay? So, yun ang use ng acetic acid. Baba na yung pH mo para yung bromine, pumasok siya into your amino acid. And paano mo naman makita na nag-move yung solution or nag-react to? So, again, kailangan ma-visualize mo siya through making your solution neutral again. Okay? So, para ma-visualize mo, need mabalik sa neutral pH. Kaya, gumagamit rin tayo again ng 5% ammonium carbonate solution. Okay, again, your acetic acid decreases your pH, ginagawa niya acidic. Your 5% ammonium carbonate solution increases your pH or neutralizes your solution. Okay? So, pag nangyari to, ayan, lilitaw yung blue to violet solution mo. Okay? So, additionally, heating of the alkaline solution would hasten the process and give you a blue or violet color solution. So next, we have your Sakaguchi's test. So here, it is a guanidinium group of arginine that reacts with your alpha-naphthol upon treatment with your hypochlorite or hypobromite under an alkaline condition. Okay, so ano yung mga reagent na gamit natin dito? Your 10% sodium hydroxide that function as a alkaline buffer, your alpha-naphthol, which is your main reacting solution, and your hypochlorite or hypobromite solution, which is your oxidizing agent. Okay, so let's explain on the next slide. Okay, so your Sakaguchi's test specifically para to sa arginine, pang-determine. Okay? So yung arginine mo daw, meron siyang guanidinium group. And you also have your alpha naphthol. So, two molecules of alpha naphthol would react with your sodium hypobromite. Okay, so in this case, your sodium hypobromite in an alkaline condition. So, you have to remember na ito, magre-react lang siya pag alkaline yung condition niya. Pag hindi, wala. Okay? So, pag sa Sakaguchi's test, dapat yan, alkaline solution siya. Okay? So, yung alpha naphthol mo, kita nyo, magiging bromine or magbo-brominate siya or chlorinate. Okay? And then, yung arginine mo, what will happen? Mako-condense siya between your two alpha naphthol molecules. Therefore, yielding a red compound or a red product. Kaya ang nangyayari, ang end result mo or yung positive result mo for arginine is a red solution. So now let's have your polys diazo test. So this is a diazotization of single cyclic side groups such as tyrosine and your histidine. Okay? So it is based on the principle of coupling between the amino acids and the diazonium ion formed in the reagent. Okay? So the Polis reagent consists of sulfonilic acid dissolved in concentrated hydrochloric acid. So the sulfonilic acid undergoes yung diazotization in the presence of your sodium nitrate and your HCl. So ano ba yung diazotization? So diazotization is a chemical process used in converting a primary aromatic amine into the corresponding diazonium salt of the amine. Okay? So ang positive result for this one, so yung histidine mo and tyrosine, it would yield a scarlet red solution. Okay, you have to remember. Scarlet red solution. So yung diazonium salts formed are then further used to synthesize many organic compounds, for example, dyes, pigments, and etc. Okay, now for your Millon's test, this naman is the nitration of hydroxybenzene ring specifically for tyrosine. So yung test na to, it is a specific test for tyrosine. Okay? So, the solution is highly acidic. So, yung reagent natin here is called your Millon's reagent. It is your mercuric nitrate dissolved in concentrated nitric acid and sodium nitrite. So, yun yung dalawang reagent natin. So, a positive result would yield a red precipitate or a red solution. So, what happens in this chemical reaction? So, you have to remember tyrosine has a hydroxybenzene ring. So, ang tawag doon, it is a activated benzene ring. So, ano ibig sabihin pag ganon? It can freely react with your nitric acid. Ibig sabihin, madaling mag-react siya. Okay? So, nagiging nitrated tyrosine and that would give off a yellow color. So, kung yellow color siya, bakit ang positive result? Red precipitate or red solution. Okay? So, to make sure na tyrosine nga siya, you need to add your sodium nitrate. And upon addition of the sodium nitrate, yung dalawang nitrated tyrosine mo, i-condense nila yung mercury. Okay? To form what? To form your mercuric phenolate. Okay? You have to remember that. So, yung dalawang tyrosine mo, mako-condense sila, and then magpo-form ng mercuric phenolate. So, that is the reason kung bakit nagkakaroon ng red solution because of that. Okay? So, pag precipitate naman, bakit nagkakaroon ng precipitate? 'Di ba meron mga tirang sodium ions. So, yun yung mga nagre-result into your red precipitate. Okay? Next, we have your lead sulfide test. So, this is the acquisition of sulfur from sulfur-containing amino acids to form lead sulfide precipitate. Okay? So, ano ba to? It is a biochemical test for the detection of amino acids like your cysteine. So, ang positive result for cysteine, magkakaroon tayo ng black precipitate. Okay? So, again, it is a detection of amino acids containing sulfur or the S group. Okay? The S group in your cysteine. Okay, so next we have your Adam-Cuick's reaction. So it is part of a biochemical test used to detect the presence of the amino acid tryptophan. Okay, ito yung condensation of your indole ring with aldehyde under an acidic pH. Okay, so when concentrated, yung sulfuric acid is combined with a solution of protein and glycosylic acid, and therefore producing a purple color. Okay, so it was named after its discoverer, Albert Wojcik Adamkiewicz. Okay, again, what you have to remember here lang is that the indole group of tryptophan, it reacts with your glycosylic acid in the presence of H2SO4. Again, your positive result, tryptophan, yielding a purple ring at the middle of the solution. Okay, next we have your Follins-McCarthy-Sullivan test. So this test was developed when it was discovered that tryptophan and histidine both produce a red color with nitroprusside under similar conditions. Okay? However, by changing the pH of the solution, tryptophan will be undetectable. Okay? So it has been stated that the addition of glycine eliminates the detection naman of your histidine. Okay? Again, Tryptophan will be undetectable by changing the pH. And then yung detection naman of your histidine, it is eliminated by the addition of glycine. Okay? So the red color is obtained after the addition of your sodium nitroprusside to an alkaline solution of methionine followed by the acidification of the reaction. So ano yung positive result mo dito? Is your methionine yielding a red colored solution. So lastly, we have your isotin test. So the detection of amino acid proline for a long time had been unsatisfactory. Okay? So methods like the ninhydrin test can only detect proline after isolation from other amino acids. Okay, so this test naman, it is the reaction between your proline and your isotin. And this is used both for qualitative and quantitative test. Okay? So, tinetest niya dito if proline siya and how much proline there is. So, qualitatively and quantitatively determining the amount of proline. So, what is the positive result for this test? You get blue spots, specifically count them. Okay?